This is the day that we're gonna have partial virtual and partial live stuff. So bear with us as we work through any kind of snafus. So the way that we have to do the virtual is we're gonna have to put a computer, this microphone down on the computer so we can catch it so you guys can hear, because the computer doesn't come over here. So anyway, if the quality isn't awesome, I apologize, but I think it'll be okay. And so we have a couple virtual talks in the workshop, and then this afternoon or later this morning, we also have a couple virtual talks. So looks like we've got more people in, and I think we should probably get going if we can. And our moderator is David Samuelson, and he's going are you going to explain how this came about?
I can.
You can? Okay, that sounds good. Okay. Yeah, okay, so if you want to use this one, you can, but then it has to go onto here when we start the talk.
Set up.
Yeah.
First talk.
Yeah. Okay, yep. Oh, maybe I should ask first. Can people hear online?
Yes.
Okay, thank you. Perfect.
Okay, yeah, so I can explain a little bit. Obviously, this is, workshop that is set up for us to learn. Myself personally, these resources are very useful, not only for research and to learn about quantitative traits, but I also use them for teaching and training for students. My department, we get people come in, graduate students, they're biochemistry and undergrads, chemistry. So they teach them the principles and concepts of quantitative genetics. Find these resources very, very valuable. And obviously, you know that they're valuable for your research because you can use these resources, community resources as tools, not only to share your data, but to learn anything from the organismal level to down to the molecular level. And glad that the conference organizers put this together for us. So we have invited people to share and do a tutorial on their resource and what we can do. And what I find overwhelming, and I think the students do too, or the researchers, when you go into this, things change and it's overwhelming to know where to start to navigate on a trait. So I try to train people to go into this, have a question before you sit down. Otherwise, you might start, learning too much or going in the wrong direction. It's important to have a direct question to start with, and it's also useful to train. You were talking yesterday to use the best appropriate organism. So this is a good way to train students, not to just to go to what they've been told, but find the best model organism for the problem. And so today, I think we're organized to learn how to navigate in the first part of this, and then the second part is designed to, okay, now you learned, you found the model organism, where can you go find the best one, and then how do you find it? But the first speaker is online, right, virtual, so it's Mahima Didi, and she's going to talk about the RAT genome database, and then we're going to have the RAT GTEX, Dave Homer, and then Laura Sado can't make it, but we won't get a talk on the gen, and then Gene Network too here. There he is, so he's here in time. Perfect. And so we'll start out with that first scenario where we're going to learn how to use these to navigate. Okay.
I thought you said, what do we have to do? Oh, Mahima, can you share your screen, please?
Sure.
Thank you.
Can you see my screen?
Yes.
It's not in presentation mode yet.
Okay. Am I audible? Okay. Hi. Good morning, everyone. I'm Mahima from RAD Genome Database, and I'm going to discuss use case 1, navigating public resources for quantitative genetics, phenotypes, and omics data for my research. Starting with the virtual comparative map or VC map, which is an innovative tool designed to allow comparison of syntenic regions across multiple species. It can be accessed from the RGD homepage from analysis and visualization or directly. There are 3 ways to begin exploring the synteny between species with VC map, using either load by gene, position, or flanking genes. Here, we are using load by gene and entering the gene symbol MTHFR. Select your background species and then add comparative species, which is human and mouse in this case. And then click on load VC map to get to the VC map results. Loading the VC map results page displays the overview and details of the comparison information at the top. The chromosome with the entered gene is displayed vertically, with the anchor chromosome, that is chromosome 5, shown in the overview and in the detailed view, arranged in increasing coordinate configuration from top to bottom. Show hide species, change the order of species, search for a gene within the current view, access previous views from the current session, and zoom the details display using functions in the top sections of the page. In VC map, syntenic blocks are aligned so that the orthologs appear in the same order for this comparative species as they do for the backbone. Since the syntenic block that contains the MTHFR gene is reversed in human compared to rat and mouse, it is shown in the opposite direction and can be viewed by hovering over the chromosome location. Variant tracks can be added from settings. In this, human, mouse, and rat heat maps show variant density. In RGD, human variants are imported from ClinVar and the GWAS catalog, and the mouse variants are imported from the European Variant Archive. Rat variants are derived from the whole genome sequencing of multiple rat strains as part of Dr. Mindy Dwinell's Hybrid Rat Diversity Project at the Medical College of Wisconsin. You can click on individual variant blocks and analyze them in the Variant Visualizer tool or JBrowse. And more information about VC map is in a poster by Dr. Tom Heyman. And for more on the MTHFR gene, RGD gene report page can be accessed directly or from the VC map page as shown here. This is a rat gene report page for the gene MTHFR. Gene report pages can also be viewed directly by searching for the gene in the RGD search bar. The gene report page contains detailed information on the gene. Click on the variants to view the damaging variants in the Variant Visualizer tool. Variant Visualizer contains genomic variant data for multiple assemblies in RGD. This is a human gene report page for the gene MTHFR. Similarly, for human, damaging variants can be viewed using the Variant Visualizer tool. Click on individual variants to view additional details about the variant, such as nucleotide, amino acid chain, polyphen predictions, etc. And each variant has its own variant page that contains more detail about the variant, such as location, transcript, and protein level consequences, etc. Using VC map, we can get more details on the gene MTHFR by going to the JBrowse tool. JBrowse 2 can also be accessed directly from the RGD tools list, and it is a genome browser and has been updated recently. JBrowse 2 browsers are available for all 10 species for which RGD houses data, and includes multiple assemblies for many. Tracks are available for gene models from both NCBI and Ensemble for easy comparison. The sidebar, which is either displayed on the left or the right side of the window, shows the track selector by default. You can expand or collapse categories in the track selector. This page shows rat MTHFR gene in the JBrowse and the damaging variants in SHRSP rat strain using the damaging variant track option. Multiple tracks can be viewed together for comparison. Here, human and rat MTHFR gene are shown for comparison, and multiple options to view synteny are available. To find quantitative data in rat for the MTHFR mutant strains, we go to the Phenominer tool, which is a tool for ontology-based standardization, storage, and mining of quantitative phenotype data for the laboratory rat. The Phenominer user interface is accessible through analysis and visualization or phenotypes and models on the RGD homepage. Phenominer has all of its selection components on a single page. The ontologies used to organize the data in the database include rat strain ontology for animals measured, vertebrate trait ontology for organizing related phenotypes within the study, clinical measurement ontology for what specific phenotype was measured, measurement method ontology for how the phenotype was measured, experimental condition ontology for recording the experimental conditions. The vertebrate trait loads automatically, but you can start with any of the ontologies by using the tabs in the lower right panel. And selections made in the bottom panels appear as chosen terms in the top boxes, and all the changes are shown in real time. Entering a term in the search box filters the list to show only matching terms. Additionally, choices made in one ontology will restrict the available option in others. For instance, selecting a vertebrate trait will narrow down the strains to only those with relevant measurement data. For example, 47878 records are there under organism system trait, including all its subcategories, while 628 records are there under SS mutant strains with a selected VT. Click on Generate Report to view the results. The results from the Generate report consist of a graph, a list of filters, and a table of result. If the measurements that use more than one unit have been selected, the graph is hidden until the user filters the measurement selection to a single unit. And data from the query can be downloaded using Download All Records. Download All Record is displayed on the pages for both filtered and unfiltered queries. Select the parameter to color the bars with the drop-down legend. Here the graph is colored by strain. The filters, the graph, and the table are all interconnected. Applying filters on the left removes the data from the table and the graph. Sort the table using the up and down arrows in the column headers. Bars in the graph are reordered accordingly. Here the value is sorted in ascending order. And the combination of sorting and coloring can highlight important results. Where data for individual rat exists, the graph shows the average as the bar with the individual values shown as red dots for each phenotype. Hover over the bar to see pop-up with the details about the average value for that data point. Under controlled sodium content diet, mean arterial blood pressure differs between SS strain types influenced by genetics, experimental conditions, age, etc., as shown in this graph. And more information about Phenominer can be found in a poster on Phenominer. Thank you.
What happens if somebody needs help using RGB? If it's like a concept of app or something, and who enters those? How would that work if somebody wanted? Okay, you're ready to respond. Yeah.
There's a contact button at the top of the page. There's also at the bottom right corner, there's a little icon that you can click and you can enter a question into that, or you can say, I like this page or I don't like this page. Thumbs up, thumbs down, so rat thumb. And those all come to, there's a number of us that it comes to, but I am usually the one that answers those questions, so you put a face to the blank hole that those questions fall into. So, thank you.
We used to have, we tried out some office hours. We really didn't have a whole lot of makeup on it, so right now in the red bar at the top of the homepage, we have this agenda and this meeting up there, but we'll put it back up. And so if you have a question and you want to have a Zoom meeting, let us know and we'll arrange it, no problem.
And will these be made available with these tutorials?
Yes, yeah, we have a,
You have a spot for that?
yeah, we have a tutorials presentations thing in the community drop-down?
Yes.
Yes, so go to the community drop-down, you'll see tutorials. Put them up on the YouTube page as well. Unless somebody doesn't want their stuff on there. If you don't want your stuff on there, let us know. Otherwise, for sure, we'll post those. We posted them from previous CTCs.
Oh, okay, great. So we're going to change gears slightly, and we're going to talk about gene expression. And I'm starting out here. And by the way, Dan, you're on the line. Dan, the person who wrote the RAT-GTEx website, is on the line. And Dan, you're welcome to jump in and unmute yourself and talk if you want to. But so I wanted to start out by pointing out that there's a project called GTEx that many of you might be aware of, but some of you may not be aware of, which is a human genetics project. And if I click on their About tab here, they will tell us that they have, lots of different tissues. So 54 different tissues. These are from dead people. They didn't kill the people themselves, the people were and then they got consent, I suppose, probably from next of kin. And they collected pieces of these people. So a number of different brain regions, but all of the different pieces that you can think of. And they genotyped those people, and that meant that they had genotypes and lots of RNA-seq data from the RNA that they extracted from the pieces of the people. And you could imagine you could then do EQTL mapping and identify in these different populations EQTLs, that is chromosomal regions, that if you inherit one version versus the other version, you'll have higher or lower expression of the gene. And they've done that with total gene expression, and I think they've also done splice EQTLs, and there've been a number of other projects. This was a relatively well-funded, I think, out of the Office of Director, and a lot of people worked on it. It turns out that there's also a group, and let's see, Zoom is like, okay, all right, great. There's also a group called Farm GTEx, which I'm a little involved with, and they lifted the browser for human GTEx and employed it for a bunch of different farm animals. And they have some nice pictures down here. Look at those really artistic pictures of different farm animals that are already hosted here. The way they did this was a little bit different, I think, and I talked with these people. They're a lot of fun. It's a great group. It's a little different in that I don't think many of these animals were genotyped on a SNP genotyping array or genotyped in the conventional sense. Instead, they just said, well, we've got all these RNA-seq reads that people had collected for other reasons. So they collected RNA-seq from farm animals. They probably did kill the farm animals because you're allowed to do that. You can then lift, you can imagine, SNPs from the RNA-seq reads, and it's the SNPs that they get from the RNA-seq reads that they're using as genotypes, and then they're using those genotypes to map EQTLs. So it's a different approach that took advantage of the fact that there was lots of RNA-seq data available for various tissues from various farm animals. And they're expanding, I think they have in mind 13 different species that they mentioned somewhere. And I should have said, excuse me for not saying, but on GTEx now, the human one, remember, they have, and actually I hadn't been here for a few months, so I hadn't noticed this. They've got what they're now calling adult GTEx, which is really the original GTEx. And then they've got developmental GTEx, and you can imagine that's from developing kids. I assume that they also died on their own. And then they've got non-human primate GTEx, which seemed like a good idea, but not a farm animal. So I guess it belongs here instead of on farm GTEx. Well, years ago, we were interested in making something, an EQTL resource for rats, and particularly focused on the heterogeneous stock rats that many of you heard me talk about yesterday, so the cross between 8 inbred strains. And we didn't feel like making our own browser, and so Dan Monroe, who's on the Zoom and really deserves the credit for this, figured out that you could just set up an instance of the GTEx browser and populate it with your own data. And so Dan did that, and it was sort of miraculous to me. I think, Dan, from us talking about you doing that to you having a draft version of it up was like 2 weeks or something. Like very quickly, Dan was able to do that. Do you want to confirm or deny that, Dan?
Yeah, the initial version
was
exactly like that. It took quite a bit of work beyond the GTEx code.
I don't mean to say that it was easy, but he quickly had something up, and then he's refined that a lot. And so that's what I'm going to show you today. So from these HS rats, we've collected tissue, and in our case, these were animals that we had explicitly genotyped as well as done the RNA-seq on. And there's a table here that you can see. I don't know if it's big enough for people to read, but there's a number of different tissues, mostly brain tissues, but we have some peripheral tissues. Some of this data is from Leah Solberg Woods, and she actually gave us some of the largest data sets of adipose and liver down here. We have a pretty big set of data from the brain hemisphere, we call it. So it's basically one brain cut sagittally, and we took the right or the left, I don't remember, but consistently one side of the brain, the other side is in the freezer, if anybody has good ideas of what we should do with it. And then a number of other more specific brain regions. There's also eyes here. Turns out we had a collaborator that was interested in eyes and made a relatively small data set. And so this allows you, the main way that I would think to interact with this is to search a gene. And I'm actually going to search SLC35F1, which is a gene that came up in a GWAS project at Montana. Laura did. There's Montana back there. I don't know if are you talking about this, Montana, or not? No, you're not. Okay. She talked about it last year. This is old news that I'm going to tell you about. Anyway, she did a GWAS that gave a beautiful result. It identified a locus that had only one gene in it, which was SLC35F1. And we knew from rat GTEx that there was an EQTL for that gene that it co-localized that was in strong LD with the apparently, the most strongly implicated SNP for that phenotype. The phenotype was delay discounting, which has got to do with whether rats wanted a small reward now or a bigger reward that they had to wait for, and the different ways that the animals made that decision. So if I put in SLC35F1, I can see all of the different tissues that do or do not have EQTLs. So it's kind of green or gray, I guess, depending on whether you have EQTL or not. We also do that for SQTLs. Dan, isn't it true that you're probably in the process of setting up other gene expression traits that are going to come from pantry? So
yeah, we've talked about that.
Now we're having a lab meeting. Go ahead. Yeah. Okay.
So haven't really done that.
Yeah, not next week, but at some point we hope to have some other kinds of gene expression traits. So splicing things and different starts and different 5' and 3' traits, et cetera, that you can extract from RNA-seq data. And then if you go down here, I can visualize the QTLs. And actually, I think I preloaded this because it was a little bit slow to load. So here we can visualize the EQTLs, and this is showing the haplotype pattern. You see there's a very obvious haplotype relationship here where these SNPs are in strong LD with each other, and these are in strong LD with each other, but they're two different haplotype blocks. And then I thought it was going to show me, on this view, I thought it was going to show me the thing with the different SNPs and their p-values. Do I have to click on a tissue to get to that?
So the one with like the violin plots?
Yeah.
I think that would be a different one. Under visualize, there's like one EQTL dashboard.
Okay.
Show that.
Oh, under visualize here, you mean?
Yeah. EQTL violin plots.
Yeah. Okay, great. So I'm not going to drag you guys through all of this, but the point is there's lots of tools that you could use here to look at the data that we have. And we have this mapped right now to RN6 and RN7, which we're going to add RN8. Don't worry, we're working on it. Somebody's winking at me in the audience about that. You can also get to the data download page. So Dan's done a really good job of making sure that you can download all of the kinds of data that you might want to get about this in case you want to take it off and do something else with it, play with it another way. So I think for the average user, the thing that this would be useful for is finding out whether or not there's an EQTL for your gene of interest in the heterogeneous stock population. Obviously, we could expand this to include other rat populations. Some people work in these commercially available outbred populations like Sprint Dolly and Lewis and et cetera. We haven't done that yet. We don't really have any data to display about that yet. And other communities, if they were interested, could do something like what we've done, what Prime GTEx has done, which is just to take this and set it up with their own data. So that is your quick tour of rat GTEx. Dan, how badly did I butcher it? Do you want to add anything?
No, that was pretty good. There's the gene search is pretty useful. I don't know if you mentioned that.
Just up here.
Yeah, if you have a specific gene that you want to look for.
That was where I got SLC35F1. And if you,
Oh, yeah.
Yeah. Yeah, there's a link to what?
When studies use the data on the data on example.
What is it? Okay. What Faith said is probably true, but I'm not exactly sure what you mean. And the downloads page, you want me to go? I see. Okay. Yeah. All right.
Original study data.
Yeah. Yeah, that's right. That's right. Exactly right. Yeah, there's a lot of different things that you can download here, like unprocessed and processed data basically can be downloaded. Okay. I think, can you search by SNPs or not, Dan?
Nope.
Nope, you can't. Impossible. Out of the question. Yeah. Yeah. So it's pretty flexible, and it is an instance of this thing that another group developed. A very well-funded group. There's a paper about this. I think there is something on maybe the downloads page that says what to cite, or is that under about now? API. Anyway, somewhere there's a thing to talk about it, and there's a nice paper describing this resource and these data sets. There's some data here that have been added since that paper came out. So I don't want to belabor this. That's the resource. We've talked a little bit with the inbred strain rat community about whether or not this should be expanded to carry that kind of data. We haven't done that yet, but it seems possible. Okay. Yes, Aaron.
There any numbers single cell?
Yeah. Right. Yes. Okay. So.
Does that get compatible with?
Yes. Okay. So I'm going to repeat Aaron's question because I'm not sure if the Zoom people can hear. So Aaron Gertz asked whether or not there was any capacity to host single cell RNA-seq data. There are single cell. We had a paper, and there are more unpublished data with single cell RNA-seq in brain regions of HS rats. So the data exists. I know that Dan and I have talked about that. I saw that on the human GTEx, they have a tab for single cell RNA-seq data. So I assume that the tool is able to display that kind of data. Dan, I don't think we've talked seriously about doing that yet. So I think that's probably would be months away. But it partially depends on whether or not there's a demand for it, right? If a lot of people started asking us for that or giving us that kind of data to host, that would encourage us to deal with it. And this is supported by that P30 that recently got funded. So this is one of the kind of resources that's supported by that P30. And so something we're trying to do kind of for the community, right? Yeah.
So I'm wondering, I mean, this is a wonderful resource, and we'd like to give you business from our GDs.
Yeah.
There are ways that we could be integrating, links or URLs
Yeah.
and everything else to this from the gene pages and the variants, variant pages, etc.
Yeah, I'll answer, and then Dan can give the correct answer. I know that there is this API thing here, so I think that would be the straightforward way for you to link to it. And Dan is a totally reasonable and exceedingly competent guy. And if you email Dan and said, this is what we're trying to do, I'll bet he would help you. Do you think that's true, Dan?
Yeah, so linking to, in what sense? I didn't quite hear.
Oh yeah, sorry. So Ann Kveitik's question was whether or not their site RGD could link to this. So I imagine they might want to say, here's a gene, are there EQTLs for it? And then somebody would click on something from her site and it would take them to RGText displaying data about a gene. Isn't that sort of what you have in mind, Ann? Yeah, she's saying yes.
Yeah, I think, yeah, you could link to the gene search results page, which if it's a valid name would go to that, or you could use the API to get data to check which results exist before linking. So yeah, there's some options for that.
Putting a gene URL. Pretty straightforward, I think, to do. Yeah.
Yeah.
Yeah. So, and just to say it one last time, this is all Dan Monroe. Like the whole thing is him. And if you have gene expression data, we can give it to Dan, and Dan has a nice pipeline to get new tissues up here. So, okay, I think that was enough for GText for one day, right?
That was great. I think that,
okay.
Do the end.
Yeah, can we all do a round of applause for Dan? Dan, and he'll be up there. Does that work? Could you hear that, Dan?
Nope, but thank you anyway.
I'll do
I
that
could see it.
All right, well, take my word for it. Okay.
Peter is going to talk about gene network. He talked about it yesterday, but now we want to just navigate.
I'll use that one. Oh, yeah.
This doesn't need to go back.
Yeah.
You need to unshare.
Oh, yeah.
Somebody needs to unshare. Okay, got it. Way. Yeah, we've got Zoom. Okay, so I've got 10 minutes, I suppose. Yeah, I just wanted to I have 10 minutes to introduce GNetwork, you know, and usually we take hours to do this. Yeah, so I'm cramping my style again. So I made no slides today. I'm just going to show you what we have quickly. But the idea is that you know, if you have your own data set, you've generated your own data, right? You can analyze it at leisure using R. You know, you can compare it with information in GText and other, you know, sort of static databases. But what we offer is that you can upload your data into GNetwork and you get a web resource, right? So you can do the analysis, which I'll show you a little bit. But the real power is in the fact that if you, for example, work on the BXD mouse, there are 10,000 other experiments in there in this database that you can compare your own experiment against, right? So you can do VR style analysis. You can correlate, you know, the traits. You can search, you know, if you're interested in a specific region or you're interested in a specific trait, you can search against all the other regions and other traits in the database, right? So I'm just going to take Rob's favorite Sonic Hedgehog. And what you actually get is a list of traits that have a significant or have a reasonably high log p value. So there's a QTL in there essentially, right? So these are all low numbers, but I don't really care. I'm just going to pick one. Let's see what it does. So you have a trait here. It's part of an existing effort. You know, it's part of an experiment. This is all described there. You can look at the trait distribution, for example. Right. So, and if you're not happy with the distribution, you know, for example, a lot of these tools they assume normality. You can transform the data. So this only has a log2 transformation. So let's apply Abe's favorite.
right? So with quantum normalization, everything starts to look like really normal. Yeah, so this makes people happy, I suppose. And then the important thing is that you can do mapping, right? So our favorite one is JAMA, which is a linear mixed model. It uses leave one out chromosome analysis, leave one chromosome out analysis, and you run it. So there's 67 samples here. It shouldn't take very long. But the key thing is you do the analysis on the fly, right? So, and here I picked to do leave one chromosome out, but you can also do with, you can do without loco. You can pick a covariate. Yeah, so now, here we see this nice peak on chromosome 1, which could be of interest. Yeah, and then here you get the SNPs that have these high log P scores. So this is a live analysis, right? So we're already unique in this way that you can, analyze your own data. And when you publish your material, you can actually publish these links with it too, so people can reanalyze, relook at the data in a reproducible way. So if you talk about fair data, fair can mean different things to different people. But I think we call it fair plus, which means you can also do the analysis again, right? Okay, so if we go back to the previous screen, this is where I started the mapping, right? There's some other tools here. I mean, you can do different types of mapping. Ignore that, but I'm gonna look for correlations. So essentially what we do here is, okay, we have this trait vector, right? And I'm gonna look in the database against all the other trait vectors of the BXDs and see which ones correlate. So you're looking for traits that are very similar to yours, or have some, similar profile, you could say. Not sure how long this will take. That was not too bad. Yeah, so here we have traits, and they, you can see they have a sample R value here of close to 1. Yeah, so you might be interested in exploring those. So you get an exploring system, right, where you can link up these traits, and we also have something you can compare to a shopping basket, so you can add these traits into a shopping basket, right? Should do it. So I've created a collection essentially, right? Now, when I have the shopping basket with these different traits in it, we can, for example, look for correlations. Yeah, so now it's going to be a matrix of correlations. On the left side is person correlation, the right side is rank correlation, and you can go visit these things. So G-network, this is just one example, but you essentially, so this is a trait correlation, right? What you see. And this is all, publishable. It can go directly into your manuscript. And so what's the real value of G-network? Yeah, it's live analysis of your data, and you can adapt it, you can share it, and you can compare it with other data sets. And there's really nothing like it. And over the years, we get feedback that people get frustrated with G-network. It's a powerful tool. It's actually a complex set of tools, and we're actively developing against it. It's like Elon Musk blowing up rockets to learn something. That's exactly how we've so far been approaching, running G-network. It blows up in your face now and then. And so, but we've actually turned a corner in this year. We have a very dedicated effort going on to stabilizing the system, to making it possible for people to easily upload their own data. It gets quality checked. There's a QA in there. And I think we, it's worth considering if you were ever frustrated in the past, Amelie, it's worth considering working with G-network again, yeah, because we'll be a lot more responsive. We've got something to build on, for everyone. And yeah, I'll leave it at that. But I think in the next CTC, we'll also try to organize a workshop, and then we'll spend more time on how to deal with this type of product. Doesn't go down well in talks, because this is what you can show, and then everybody shuts down, right? But yeah, so if you have data and you want to, you want to publish it in some way, you want to write your manuscripts, consider doing it with us. Couple.
Which all the compute constant, is it all in Memphis or is it a commercial cloud or how do you do that?
Yeah, so we run our own machines. We have about 30 of them at this point, and we also do the Genomics on those machines. So it's pretty hefty setup. Yeah.
And then what do you say change for that? Right? To maintain something like this costs money, develop costs money. So how have you?
Yeah, so the development team is quite expanded. I think we have 10 software developers actively contributing. A lot of them are, yes. Yeah, they wouldn't work otherwise. Quite a few, a team is based in Kenya. Yeah, so over time, Genetwork has been funded in different ways. Currently, we're mostly NIH funded, some NSF component. And we're trying, to continue in that vein. Yeah, it's very hard to do something commercial. Or, I mean, we thought about it, but in the end, nobody's willing to pay for software, unfortunately. Yeah, so but I think the NIH is also the institutes are getting more and more interested in supporting this type of effort. And so it's actually not a bad time. But I think when it comes to your work and the rat genome database and Laura Saba's work, we should definitely look at ways of collaborating. That's your question?
That was my comment. We have all these direct people to interdirect, we need to be more interconnected. Really helpful. So if there's any way that you have suggestions and do you have templates for people for their data sets? You have
Absolutely, yeah.
and standards like I saw a trade and I'm not sure what that trade was.
Right.
What's that trade, that number underscore?
Yeah, so there should be metadata that's posted with it, right? So metadata is a topic in itself because people somehow are strangely reluctant to provide metadata. Yeah.
We're going through geo and I'm assuming this is all expression data. Is that what that trade was?
Yeah, this one is, yeah.
Okay. And with the RS information, so we're trying to make sure that you guys can all get the SNP IDs, right? The RS IDs. So we have a big release that should be out. It was at EVA for quite some time, so it will be back to us very soon. So we'll distribute that as much as people can.
So, this may be a totally naive question, but how do you handle the X chromosome, and can the users of your website look for sex differences in this, for example, varying this correlation between males and females? Can they do it easily?
So 2 questions. Yeah, so and I attended your talk and I enjoyed it very much. For sex chromosomes, unfortunately, the support is pretty poor, right? So, and this is historic. I think we ought to change that. When it comes to what was the other question again?
Can I look at your website and see this for males?
Oh, okay. Yes. So we.
I don't think that mic is on.
Okay, so the other question is, how do we handle sex, right? So in general, we handle sex as a covariate, right? So it is actually in the database, and you can use it to split out your analysis, if you will.
Numbers take sex out of analysis.
Yeah.
Okay.
Yeah.
I want to put it in the analysis.
It's an interesting one. Yes. We can talk about it. I think ultimately it's a malleable system. So. One more from Abe.
So I think it's great that you do all the computing on the fly. I didn't say that, but for rat genomes, we have the opposite approach, which is that things are all pre-computed. So it's just pull it up. How long does it take to do a trait that might have hundreds of individuals and, 100,000 markers or something like that? Is that a job where I have to go make a copy and come back?
Yeah, so when we did your 1,500 individuals, it took too long, right? I mean, we were.
Maybe years ago, but I wonder what.
Yeah, so 1,500 is probably still stretching it, right? Because we tend to focus on tools like Gemma, which are actually meant for smaller populations. If you use a larger population ones, you have to, there are the other trade-offs that you make, right? You lose some information in effect. Genomics, also part of the gene network effort, has created bulk LMM. Yeah, so which actually scales in the direction of phenotypes, traits. And so the number of individuals is always a quadratic issue with large linear models. And so and the way the people speed it up, if you do human style cohorts, you actually do a linear regression first and pick the ones that are significant, and then you do the LMM based on those, so you get a subset. In the process, you absolutely lose information, which is fine, for human. Who cares about human? It's just big mouse. Yes. Yeah, so definitely when you upload your data, it's private. Yeah, so it's, you can assign people that can access that data yourself. So it's all built around groups, PIs essentially. Yeah, so and then at some point you want to publish it, you put it in the public domain. It's just a click away. Yeah.
Sorry, so that means that you're storing like all the analysis for people citizens. Or you're trying to like so it wants how long you store them for. Yeah.
The analysis? We don't start analysis per se, right? So when you do analysis, and I didn't show that, you get a link, which points back into the stuff you've been doing, right? So you can destroy your track record also if you want.
Okay, that's not important. I'm shocked that you're storing people analyze data from them.
Yeah, we are not. We don't track people. I'm actually very much on privacy myself. I don't use a standard Android phone. I don't like to use Google products. I don't use Microsoft. And so it's a long list of things that you don't do. But you appreciate also the other people, they may have the same interests. But anyway, I'm around all day, so
A little ahead of schedule because we had one fewer talk. So you want to take. So we can have 10 minutes to chat, or you can take a quick bio break and we can get that together. If not, I don't have a schedule from me. Okay, so anybody have anything they want to talk about, or you want a 10-minute break? Sounds like it's a break. All right, we'll be back at 9:30 to start the second use case.
Okay, we're gonna get going again. I apologize for online folks. We got really into talking, so we're a little late grouping back. So we're gonna continue on with our second use case. Can you confirm? Can someone confirm you can hear us online?
Yes.
Excellent.
Yep, sounds good.
Though if you're not directly at the microphone, sometimes it gets a little bit garbled, so.
Yeah, okay. So the online stuff, Cindy, we have you with a different microphone right on my laptop here because this is the way we're having it work. So hopefully this will work okay. I'm glad that I heard your voice. And could everyone hear? Okay. All right. So we're continuing on. And David, are you gonna? Okay.
So the setup for the next part of this workshop is you queried, you navigated, found interesting information for your quantitative trade, how you want to see what models are available out there. Right? That can be important to know. Well, maybe the model hasn't been made and you need to make it. But obviously, as we talked about yesterday, picking the correct model organism and lots of resources, not only mouse and rat, right? We have things for sleepy fish, flies, a lot of resources. So now this is going to be directed at how do you decide what the best model is. Eventually we'll get into where you find it. So Cindy Smith is going to talk about mouse genome informatics. And once you have been to that website, there's a ton of information on there. So it's going to be interesting to see how she can help us get over that overwhelming stage and find the correct model. So, Cindy, are you ready?
All right.
All right, we can see that.
Okay, can you see the full screen now?
That's not your presentation.
The full presentation? Yeah. Okay, thanks. Hi, everyone. Good morning. I'm here to talk about mouse genome informatics, which is a worldwide database resource for data about the laboratory mouse. We provide integrated data about the genetic, genomic, biological function, phenotype, disease model data to facilitate the study of human health and disease. Like Dr. Samuelson said, I could go on and talk about this for about 2 or 3 hours, but we're going to focus on a simpler question today is how do we find mouse models of disease? And for this, I will be focusing on 3 parts of our database, our informatics resource, the quick search, the human mouse disease connection, and the international mouse strain resource, which can all be found and accessed through our homepage at Mouse Genome Informatics. In addition to that, we have advanced query forms that I'm not going to show today because of time. There's MouseMine, which is a customizable query access to MGI data as well as our APIs. We have a series of downloadable reports, as well as JBrowse and the multiple genome viewer to compare genomes among strains and SNPs. But to answer the simple question today, how do we find mouse models of human disease? I'll start with the quick search. So if we type in, you can type in keywords, symbols, and IDs here. Any gene name, any vocabulary term, excuse me, allele symbol can be entered here. And you get a summary results in a tabular format. So the first tab includes genome features, which is genes, other genome features such as like promoters and regulatory regions, QTL, etc. There's a second tab of alleles I'll show you in a moment, vocabulary terms, strains, and other results. If you search by an ID or set of IDs, they'll come in that tab. So if we search for obesity and we find all of these genes, that this is a disease ortholog, and we want to know a little bit more about that gene, we can click on the gene symbol and go to the gene detail page where we can find out about the human ortholog, human diseases associated with both the mouse and the human genes if you click on this More tab. And you can also get a listing of all the available strains or lines that carry a mutation in the ABCA1 gene if you click on this link here. And just as a preview, I'll talk a little bit more about this. You click on the link and we have over 90 strains that carry a mutation or ES cells that carry a mutation in that gene. that you can then click on and order from the appropriate repository. If we go to the next tab of the quick search results, the alleles tab, likewise, if you click on the allele symbol, it'll take you to the allele detail page for that particular mutation. It gives you a lot of details about how the mutation was made, what the strain of origin is, links to the Alliance of Genome Resources, and also to the IMPC if this was an IMPC gene. And to focus a little bit more on the phenotypes and disease model sections, you can click on any one of these toggles or check marks to get more details about the phenotypes, or you can click on the view phenotypes for a concatenated display of all of these, all of this information. If it has been annotated as a disease model, it'll show up here, in this case, obesity, and you can click on the genotype for more details. And then finally, if this mouse is available, if this particular mutation is available as a mouse strain in any participating repository, the links to the IMSR, the International Mouse Strain Resource, are available here. So there's 2 strains carrying this particular mutation, or 17 strains carrying any mutation in this particular gene. If we go on to the vocabulary terms tab of the quick search for obesity, we'll find that we actually have a disease term, obesity, but there's also other vocabulary terms of interest here, human phenotype, as well as the mouse phenotype of obese that may also be of interest. Not all of the mice annotated to the phenotype term obese have been annotated to the disease obesity, because sometimes these phenotypes are involved in other diseases that have been annotated, such as the ones you see down below. If you click on the associated data, such as the mouse models or the genotypes, it'll take you to a summary view of those genotypes or mouse models to peruse. So if we click on this one, it'll give you a summary view of all of the models, and this goes way down the page, of all of our mouse models that we've annotated to the term obesity. This particular browser comes in several tabs. There's a term browser, which gives you information about the term and its relationship to others in the ontology, the listing of genes that have been annotated to this term, derived from these models that I'm showing you here. They're downloadable, either an Excel or text file, and gives you the genetic background that these are studied on, because sometimes phenotype and disease can change if you change the genetic background, as we all know. The reference that supports this annotation, and the related phenotypes. If we click on the phenotype view, it gives you a summary of the phenotypes that are associated with this particular genotype, as well as mouse models of disease, in this case obesity, as well as type 2 diabetes. And it also tells us from the IMSR that there are 2 strains available that carry this particular mutation, and 120 that carry any mutation in LEPR. Another interface that we use frequently to identify models of human disease is the human-mouse disease connection. It starts with a simple Google-like search, where you can select a field of the type of what you want to search for. We also have various other downloads and useful links to some of our downloadable reports, as well as external links, the Matchmaker Exchange, which helps clinicians and researchers connect through exchange of phenotypic and genotypic profiles to help clinicians find researchers working on particular mutations or genes that are of our interest to clinicians. The human phenotype ontology, from which we derived our human phenotype annotations, as well as the disease ontology, which is the ontology that we use to annotate human disease, as well as most of the Alliance of Genome Resources members, including RGD. So if we select the disease or phenotype name and then start typing, it'll give you an autocomplete with terms that you can use to search, and you can just click on that, and you'll get a result set, which again is in tabular format. There's a gene homologue by phenotype diseases grid result form for a quick summary view. There's a genes tab and disease tab that I'll show you in a moment. In this, we have both the human genes and the mouse genes, and you'll see that not everything is always a one-to-one orthology. Human has only one AGTR1 gene, whereas mouse has a split to 2, but we've grouped those together in an ortholog cluster for display purposes. The human phenotypes that we've derived from the HPO are shown in orange. Mouse annotations are shown in blue. If a gene has both mouse and human phenotypes for the same summary system, is shown as a diagonal grid. The intensity of the color indicates how many annotations are present. So the darker blue indicates that there are a lot of annotations for this particular cardiovascular system for this gene in the mouse. And likewise, for the human, there's a lot of annotations in the vision eye system for the AIPL1 gene. And if we actually click on one of these squares, such as this one, we get a summary and comparison view of the human phenotypes for this gene, that Alström syndrome, which includes these phenotypes, and the associated mouse genotypes and their phenotypes. And if you see one that you think is of interest, you can click on the Find Mice tab, and it'll tell you that for the ALMS1 BBB mutation, there are 2 strains available that carry this specific mutation, or another 150 that carry any mutation in that particular gene. The second tab of the human mouse disease connection results for obesity is a genes tab. You can download this and play with this. Almost every column is sortable. There are also filters that you can apply, such as a filter by feature type, whether you want protein coding gene or transgene or any other kind of limitation. You can filter by the actual genes, or you can filter by phenotypes and diseases. So all of the resulting genes here are a combination of obesity for the actual disease, or the term obese and phenotype. So if you wanted to limit to just the obesity disease terms, you could do that by the filters. In addition to the gene symbol, human disease and phenotypes, we also list all references associated with those disease and phenotype annotations, as well as mice mutations in this gene. And this link will take you again to the IMSR. The diseases tab, you can sort by specific diseases. We present their ontology IDs, as well as the related gene in OMIM, the related record in OMIM. We provide a link out to all of the mouse models and the IMSR. For each disease, we list the associated genes from mouse models that we have annotated, and the associated human genes. The source of this information is from the Alliance of Genome Resources, which includes a variety of resources, including OMIM, Orphanet, et cetera, as well, ClinVar, as well as annotation from RGD. And again, we include references for all of these assertions that you can check. Now, getting back to the International Mouse Strain Resource, this is essentially just a worldwide catalog of repositories, unified catalog that includes resources in North America, in Asia, Australia, and Europe. You can limit your searches to specific regions if there's a location or geographical barrier to obtaining resources from other areas in the world, or you can, limit to a specific repository such as MMRC or JAX or any of these other places. And you can limit by strain state, including ESL or live embryos or cryopreserved materials, and any kind of strain type, as well as the mutations that they carry. If you want a list of all of the repositories, you can click on our repositories tab and it'll tell you the listing of all of the currently participating repositories, the last time they deposited an update to our resource, as well as a downloadable file of all of their strains and contact information. You can search by gene or mutation in this quick search box. And if we search by one of our genes that we showed earlier, ABCG1, you can retrieve the 53 strains that carry a mutation in that particular gene. You can also filter by the state, as if you didn't do that in your initial search. So if I don't want the ES cells, I just want cryopreserved material or live mice, you get a summary list here. In addition, you can link directly to the repository strain, as well as the allele record in MGI or the gene record in MGI to get more information about these mice. But clicking on, as well as you can email the repository or go straight to their order form. And you can also link to the repository strain data sheet if you click on these links. And it'll take you directly to example to the MMRC for this particular strain, including all of the information as well as ordering information for this particular strain. Thanks. MGI is a huge resource and it takes a lot of people to bring this to you. In particular, Howie Motenko works on the IMSR, as well as Michelle Perry, who helps the repositories contribute their information and tries to help them with their nomenclature, as well as all the other curators who participate in the annotation of disease models in MGI, and the software engineers who help bring you that information via web and other ways. So I will stop there. Thank you. Are there any questions?
In that the mouse human gene connection, what's the other space? Just based off literature for the human gene list, like published literature, or do you have GWAS results that may not be published in the literature in making that gene list for the humans?
Cindy, can you hear?
No, can you repeat the question, please?
Yes, so
If you come closer to this, then she can hear you through there.
okay. So in the human mouse gene connection, that's a powerful tool. I was just curious, and is the human gene list, is that evidence for that? Is that just literature based, or do you have GWAS results or other evidence going into that to make that list?
Yeah, so we it's a combination of things, and mostly it's derived from the Alliance of Genome Resources, which I think is talking here too. It's curation from some from RGD, some from ClinVar, some from the Human Phenotype Ontology Project, and I don't think we're incorporating GWAS results outside of that. Jennifer, do you remember? I think that was it. And OMIM as well.
No, I think that's it.
Yeah, yeah.
More questions?
So things where there's definitely some evidence, published evidence.
All right, thanks again.
Thank you.
Jennifer Smith is going to talk about the rat genome database. I'll use that to find models.
While she's loading up, I just wanted to give a quick note. The reason that we have this use case was it was submitted by one of our graduate students, Mark, back here, who is going to take anyone who's going to listen to Carol this evening, but I asked some of them to give us use cases so that we can make tutorials. So we'll continue. Thank you.
Okay. Can you hear me? Yes, no, maybe?
I can hear you in here.
Can those online hear me?
Yes, we can hear you.
Excellent. Okay. So as was stated, my name is Jennifer Smith. I am the scientific project manager for the rat genome database. And I have to give some credit to Mary Kaldunski. She was the one who put together all the slides. So I'm the talking head up here, but she's the one that actually did all the work. So, nope, can't do that. Hang on. I'm gonna do that. No. Wait. Okay. So there's two main ways that we're figuring people are going to come to RGD to try and find a model. One is to have a disease in mind, and the other is to have a particular gene in mind. So I have 4 different methods that I'm going to try and make all the way through to show you ways that you can come in with one or the other of those possibilities and actually find what you're looking for. I will mention, no, wrong way. I will mention that we have a Find Models portal, and I am not going to talk about it. Mary has a poster, so please check her poster out. Check her poster out. She has lots of information about it, so we decided that we would stick with the other methods in RGD and not talk about that Find Models portal. I'll mention it once or twice, but I'm not gonna give you any details. So method 1, come in using the keyword search here. It's the search bar at the top of most RGD pages. It's simple, it's straightforward, and it returns results in multiple different categories. So if you enter obesity and you click this magnifying glass here, then it will open a results page, and that results page will show a categorized table of data for the term. And I just want to mention that this is a keyword search against the gene symbol, the gene name, a description of a strain, those kinds of information. It doesn't actually search the ontology annotations. So I will get to how you find things using ontologies in my next method. But for this, it's actually a keyword search. So this search result has has all of the, yes, all of the data types going along here, all of the species going along there. And each of these is, a number for the number of genes in rat that came up as being somehow related to obesity. And here you can see that there are 6 strains that are somehow related to obesity. And you'll find in a later slide that we actually have more strains that are annotated to obesity as a disease term, as opposed to just the name of it has something obesity in it, or it's described in the definition as a model for obesity or something like that. So if you click this number right here, then it opens the search results for obesity in rat strains. Each strain symbol is clickable and will open the rat strain report page. If there is a blue PM icon, that denotes that we have quantitative phenotypes for quantitative phenotype data in Phenominer. And I'm not showing it here, but if there's a purple VV icon, it denotes that there's variant data in RGD's variant visualizer for that particular strain. So the strain report page is the primary comprehensive location for any information in RGD for that strain. So you've got the official strain nomenclature, you've got a strain symbol, there may or may not be a name, there's a number of persistent identifiers that you can use to search RGD. To find information, there's a description that usually tells you something about how the strain was developed. There are manual disease annotations and phenotype annotations. And I'm just going to mention, there we go. I just wanted to show that if you, the original view of that was a summary view, but if you open up the detailed view, you've got some additional information here, not just the name of the disease, but that this shows susceptibility to a particular disease, or it's a spontaneous model. And then there's information over in the notes that say what it might be compared to. So in this case, it's compared to the wild type litter mates. In some cases, it might be one strain compared to another, that you've got, this one shows obesity as an obese rat, and that one is a lean rat, and you're comparing the 2 strains. So in addition to that information, then there is information about the Phenominer quantitative data, which I'm not going to talk about because Mahaima already did. And then related QTLs, so any QTLs where this strain is one of the ones that was in the cross will be listed. So method 2 is the RGD disease portals. So these organize information for the most highly annotated disease categories. So there's actually 15 disease categories. And since we're talking about obesity today, we're going to look at the obesity and metabolic syndrome portal. So on the disease portal page, there are toggles or buttons to alter the page information to be focused on what you might be interested in. So there are options for disease, mammalian phenotype, human phenotype, if you're looking at human, biological processes, et cetera. I'm not going to read through the whole list. So if you click on one of those, so you say you click on mammalian phenotype for rat, then all of the information on the page will change to the information for the phenotype that you choose, instead of for a disease specifically. And we have 10 species currently in RGD, and you can look through the information for any of the species. For most of these, the diseases are based on the curation that we've done for human and rat, rather than for most of them, we don't have the bandwidth to be able to actually do the annotations for that species specifically, but we propagate the annotations based on orthology from, say, human to pig or human to 13-lined ground squirrel, which is a model for eye disease, but is not used a lot, and there isn't anybody that does disease curation or gene curation for the squirrel. Uh-oh. I think that was interesting. So the default ontology, so I don't know, does everybody know what an ontology is? It's a structured vocabulary that gives you information of, the more general terms have child terms that are more specific, usually going down. So the default ontology that's displayed is the disease ontology. And the page default, the default term that comes up for the page will be the least granular term that fits the portal category. So in this case, since we're looking at obesity and metabolic syndrome, the most general term is nutritional and metabolic diseases. And for nutritional and metabolic diseases, we've got a number of genes. You can see here that you get a list of genes, a list of QTLs, and a list of strains for rat specifically. If you're looking at other species, it'll be mostly genes. But we have a list of 204 strains for that top level term. If you navigate down to the specific term obesity, there are actually 32 strains, which it shows over here. And I should note that these include not only strains that are specifically models for, in this case, obesity, but it also includes strains that are used as controls for those models. So both of those will come up. You can click on a gene, QTL, or strain symbol to access the appropriate report page. So if I click on that F344 LEP M1 Kyoto strain, I go to the strain report page, and as mentioned before, the strain report page is formatted to provide access to all of the information. So I'm not going to go through, since I already talked about what information is there, but I want to point out that one of the things that is on the strain page is, no, wrong button again. Okay, sorry. Sorry. Nope. There we go. Sorry, got myself lost. Okay. So one of the things I wanted to point out is that we have information on the strain page about where you could get the strain. That is the most important piece of information on this slide. Also, the annotations include links to the related references where that data came from. So if you want to know the data provenance, you could go to the reference report page. That shows all of the annotations that we got out of that paper, not just for the strain that you used to actually find the paper. So here, annotations were made for insulin resistance and obesity to the strain, the allele, and also the rat gene, plus the annotations were assigned to the corresponding human and mouse genes based on orthology. So going back to the disease portal page, in addition to the strains given in the interactive list, there's a list at the bottom of the page. So it's circled there to RGD's Find Models tool that I mentioned before. And just to give you a little preview and to remind you, go see Mary's poster. So method 3, search for strains using a specific disease or for the intersection. So if you want to say, I want a strain that is not just a model for obesity, but is also a model for diabetes. This is the tool that you can use to do it. It's the Object List Generator and Analyzer, or OLGA tool. So from the Analysis and Visualization menu in the top black bar, you can choose OLGA, or you can use the prominent button that is there on the RGD homepage. So the default when you go into OLGA is to search for a gene, but you can use the drop-down to change that and choose strain. And then there are a number of search options, but we'll look for ontology annotations since we're interested in obesity. So we'll choose the disease ontology. We'll start typing in that search box, and autocomplete options are provided. So we'll choose obesity and then click continue. And the result will be a set of, The results set shows the same 32 strains found on the disease portal page for obesity. And then from here, you can wow. And when I hit it, it goes way far. Anyways, from here, you can either analyze or you can actually add another list of strains. So to do that, we'll select add another strain list, bring back the search options, and enter diabetes in the search box. And the tool will give us a preview of the 88 strains that match our, and this is an ontology search, match our search for the term diabetes mellitus. And then it gives you options right here for how do you want to combine the two lists? Do you want to join them together? Do you want to do a union? Do you want to do the intersection? Or do you want to subtract the second list from the first list and have obesity but not diabetes mellitus? And so we're gonna choose do the intersection, and we'll get the list of 6 strains that are annotated to both obesity and diabetes. You can see here that you see the original list, you see the second list that we added. You have the option to, I didn't want to actually do an intersection, I wanted to do a union. So you can change how you combine them. You can remove one of the lists, you can remove individual strains from a list, and then it will give you the results based on those selections that you made. So method 4, almost done. The fourth method is to use a gene to find a model. So if you're starting with, I have a gene I'm interested in, I want to know if there's a knockout rat or a rat that's genetically modified for my disease or for my gene of interest. And so to start with a gene of interest, you enter a keyword. You can do the, you can either do a specific gene search. You can also actually put it in the general search that I talked about earlier, but we'll talk about the gene specific search. So you can put a keyword into this search box, and hit search genes. And then you'll open up result page that gives you the list of genes that match your search results. And you can click this gene symbol to access the report page for that gene. the gene report page is formatted pretty much the same as the strain report page. Entries in the menu down the left sidebar will jump you to different sections and different information organized by category. There is a list of genetic models that comes up at the top of the page. So if you're interested in models where that gene is genetically modified, there's a list here. There's also, you can jump down the page by selecting related rat strains or genetic models in the left sidebar to move down to that region on the report page. And then it will show you the results in those two categories. And you can click on a strain symbol from that list to open the relevant strain report page. And I'm not going to go through the report page because I've already done it twice. So that's it for me. I didn't put together an acknowledgement slide, but I obviously have to acknowledge everybody from RGD because it's the most incredible team of all the model organism databases. I'm totally not biased. Aaron.
You'll have to repeat that.
It's going to be possible to put a link to that G-text right here because now you can find the HS strain where the leptin receptor is down-regulated or upregulated, right? Without having to go like it is cross-linked.
Yes. So the question was, can we cross-link to rat GTEx from our gene report pages? And I believe we decided that the answer to that was yes. So.
The URL says the gene name, so that should be right, John?
We're not going to put our developers on the spot this morning. Any other questions? Well, I was trying to look for the model for my favorite gene, and I don't see those things here. So I think it means there is no model. If there is no model for the gene, it will be visible. Is that right? Correct. Okay. Yes. The question was, if there isn't a model for the gene, does that mean that they won't be there? Those sections won't be there, and the short answer is yes, but
it's only if it hasn't been curated.
No. Well, okay.
It has to exist in a paper or through the RRC or.
Well, okay, yes. If
it's just a model that somebody's using in their lab and nobody knows about it.
Well, I think that's a really important point. Like, it's not having nothing there, right? I think having something that says no model is possibly curated in RG relative to this case. And possibly, right, linking to another page that says how, maybe links you to transgenic scores that can help you make the model.
Yeah. Yeah. Right. Yeah. Yes. Yeah. I think that's a great idea. The comment was, instead of just not having anything there, maybe have something there that says, we don't have any information on models for this gene, genetic models for this gene, and linking to places that can help you make a model. Maybe have something there that says, if you have a model and you're not seeing it here, or somebody who has a model and you're not seeing it here, then you need to let us know. We do try and search the literature on a regular basis to find new models, but it only works as well as it works. So.
Unfortunately, we do have a collaboration with Elizabeth, so that if anything's deposited in the repository, she sends them our way to name the strain, so then they're registered, and then we can put them on the page. So that's at least some cross-pollination, but I think that's a great idea.
Yes, and the other thing is, sometimes strains are registered with us, but the researcher asks to keep them private. So just because it's not on the page today doesn't mean it won't be on the page tomorrow if they tell us that we can release it. So there's that too.
I am indeed.
Can you use that one?
Oh yeah, Wendy? We already have it linked to GTEx on our gene page. Or did you just put it up there, Akil, while we were sitting here? At human or rat GTEx? Awesome.
We're better than we thought we were.
So if you look at, that's on the gene page? Yeah, so if you look on our gene page, we have a link out to rat GTEx. All right, I know we're running pretty far behind on time. I'm going to try to make it up, but feel free to interrupt me. Let's see, I think can you hear this okay?
Yep, you're great.
All right.
You can use the clicker to switch the slides or the mouse.
The mouse works too?
They both work.
Yeah, but they can't see this. Oh, okay, I'm going to try this. I'm going to try using the laser and I'm going to go back and forth and I'm going to stay right next to this so everyone online can hear as well. So today I'm going to talk a little bit about the Alliance of Genome Resources. I sort of mentioned it before, but specifically to look for obesity models, and the URL for the Alliance is alliancegenome.org. I encourage you to go visit it. So the Alliance is really meant to be a comparative genomics knowledge base, and it really is a conglomeration of mouse, rat, zebrafish, Drosophila, C. elegans, yeast. We recently added Xenopus, two different species of Xenopus, and then of course humans. So we really want to bring this back to the human condition, right? That's really the point of using our model organisms. And so what they've done is the model organism databases here have basically harmonized their data. So they found where there's common elements and they've put those information in one standard form, in one standard place so that you can search across species using sort of the same search space, right? There's information so far on alleles and variants, on function, phenotype, human disease associations, expression, interactions at molecular and the genetic space. And the way you get into this, when you go to the homepage, this is basically what you see. And so it's a search box. And sometimes it's confusing. I get confused all over the place. When you put things in these search boxes, it's like doing a Google search. So it's not necessarily looking at one specific thing. So if you get a different number from this or a different information from this general search, it's just because it's like going to Google. And people say, can we download this? Well, can you download all Google? I always get pushback from our developers on it because we always want to download that information. It is a place that you start. It allows you to just put in a general search, or you can look for specific categories, and I'll go over that. Don't do it, but if you put nothing in the search bar and you click search, it brings you back all of the information that is currently in the Alliance of Genome Resources. This number is ginormous because we have all of the human variants. However, it is extremely expensive to be holding those, and most of them have no information connected to them. So we've made the decision at the Alliance to only bring in ones that have some function. And so RGD is donating all of our ClinVar variant annotations to the Alliance, and those, I'm not sure when they'll be up, but they'll be up really soon. So keep an eye on it. There's still about 40 million, I think, in here, but between the time I made this slide, that changed. So there's also genes, models, gene ontology. So the GO consortium is part of this. So all gene ontology annotations are in here. This HTP data set index is always confusing to me. It means high throughput expression data. It's not variant data, but they call the variant data high throughput variant data as well.
And it's just the metadata.
It's only the metadata,
Not the,
right?
the actual.
And then diseases. So you can see there's a pretty good conglomeration. There's a lot of genes because there's a lot of species, right? And so it's not species specific here. You can get to it several different ways. You can go into the website as users, that's where I go, but there's also downloads, bulk files, there's APIs. And I will say that our goal is to be releasing the information on this every 4 to 6 weeks. In general, so far, there's been about 6 a year. If you scroll below the fold, you'll see sort of the goal of the alliance and the organisms that are in there. There's also a join the community. So if you want to get on the discourse, you can join the community pages and find out what's going on. Okay, so now I'm going back to the search box and I'm putting in obesity. And here is, again, these categories are kind of nice because it just gives you an overview. But it allows you to see anything that has obesity in a description anywhere. It's going to come up highlighted, and you'll see that there's 2,200 over pages of obesity annotations, and there's 113,000 results. So that's too much for me to look at. So I am going to then you can look at, refine the search by looking at, for instance, disease here. I can also just click here, go to obesity, and this will take you to the obesity page, disease page at the Alliance. And this again has a sidebar so that you can navigate. There's a summary that tells you basically what obesity is, although I think we all know that. And some of the child terms and so forth, where there are annotations, and it turns out there's annotations for most of the species that are in the Alliance here. If you scroll down a little bit, what's really nice is you'll see all the associated genes for obesity, and this is across all the species. So here, if you click on this, you can see all the species, and you can limit by species, or you can look at all of them. So it is a way that you can filter through what you're actually interested in seeing. So those are genes that are associated with obesity across all those species. Then if you go down to the alleles, you have the same basic format, and you can, for instance, here screen only for alleles from the mouse. This would be sourced from MGI, and it has the relevant information. You can click on this and see additional details on each of these alleles that you can find through the curation from MGI that's been put into the Alliance Central. And again, here's models. So there are associated models down here. And here, there's a bunch of different rat models that have annotations to obesity. So it's just a nice centralized way to look across. Now I'm going to come back to this page, and I'm going to look in obesity and go to the genes, because there are gene pages. There's 3,450 genes that have some association with obesity. And here I get to, here's Drosophila, here's mouse, and here's C. elegans, and they all have some sort of annotation to obesity. Who knew, right? I'm gonna follow up with the ApoE for the mouse and go to the gene page. So this is the gene page, and again, it's got a lot of information down the sidebars that you can scroll down. So the sidebar jumps to all the information. And in this, on the gene page, you automatically get orthology information for all of the species that are in the Alliance, and they give you some sort of confidence on different algorithms and calling those as being orthologs. There's also paralogy. So if you're interested within your own species, you can look at paralogs in there as well. Down here are models, because that's what we're looking for, are models of obesity. And here's a whole list of different models and their associated human diseases. And then any other phenotypes that are in there. And we only show a certain number, but you can expand that out and go see more stuff. So maybe I just want to look for models. I don't want to just do a search for obesity. So now I can look for model obesity. And I'll just get models pages, and I have 491 models results for obesity. Here are the ones by strain. So there's plenty of them for mouse. And I won't go over this because Cindy went over all of that very nicely. If you want to go to rat, you can click on there and it will show you the various models have been put in for rat. And if I want to look at this leptin receptor mutant on a Sprague belly background, I can look at that. And actually that will link us back to the RGD page with all of the information that Jennifer showed. I can also go back and look at zebrafish. I didn't know that I always asked, how do you measure fat in a zebrafish? But apparently you can. And so these are the zebrafish models that have some annotation to the disease as obesity. And again, if you click on this, you will get taken to you'll be taken to ZFin and get some additional information, including this particular ZFin. I don't know if this is a strain or if it's it's a strain or it's a genotype.
No, it's a fish. Or it's a fish.
Okay, it's a fish. So you should be able to go to ZFin and order up that fish if you want to look at this one, right? And then this actually shows that this has increased food availability, and it shows the citation. So you can go out. So there are links back and forth, which is kind of nice to be able to directly go to one place and then go to the different models. As a summary, I know I went really fast, but I'm trying to sort of get us back on time. There are a lot of downloads on the Alliance, so you can use static file URLs. They're updated with different versions. This is a version of Alliance, I apologize. There's different formats that you can go in and get different formats, JSON files or TSV files across all of the different organisms or expression or different data types that are in there. And this is the team. So I just want to give a shout out to the whole Alliance team. This has been an interesting and fruitful collaboration. And it was really interesting to bring these different model organism databases that have been around for decades together into one spot and to harmonize this information. And it continues on. So I don't have frog on here because this slide was made before, or the picture was made before the frog came in. But this is funded by an HTRI. That's it. Happy to take questions. Nope. There's no questions. We can move on to Abe, right? Oh, yeah, and it's break after Abe, right? Oh, you have a question? Yeah. I know. Like I said, yeah. Oh, but you had a question, David?
Well, it might be too specific, but for a human gene, I mean, I really like how everybody, when they incorporate the human and somehow even make it as highlighted as you can, because people go into these sites, that's what they're going to be looking for, and that's what will get them into a model. So I was wondering the human gene to be associated on your list. If it comes from GWAS, a lot of times the genes aren't really known at those sites. Do you include all the genes that would be annotated in that risk allele?
So in the Alliance, we do not have GWAS data in there. We'll be putting the ClinVar annotations in. So we will have human connections from the genes that way. I don't know who's talking or if there's a poster on this. Yeah, Jennifer has a poster. We've recently brought in the human GWAS information into RGD. And because we do those connections, we're really trying to connect those loci up to, we're working on it because we need more rat GWAS data.
But it's connected up as far as we can.
Yeah, I know. Oh, you're on my radar already.
Is that the damn?
Yeah.
There, it's working for me. Has this been working for you guys? Can you hear this? Oh, this is the dead one, maybe.
No, it should be okay. Let's just turn it up.
Where's that? I see.
There
Okay.
you go.
Someone told me not to blow on this. What? Oh, yeah, well.
Did you make rat names for them?
We did.
All right.
Yeah, Jude Paw, that's right. Okay, so this is something I had mentioned yesterday, and but that's because I didn't realize that I was on the agenda to talk about it today. So Radica is this thing that we have been doing, and actually we've been doing it for more than a year now, but we're sort of officially doing it now. And the idea is that we are using previously collected data where we know about phenotypes and we have genotypes on the animals, and now we have new animals that are born in our colony, and our colony in San Diego. We get about 500 animals born every 3 months, and most of those are going to ongoing GWAS projects. But before we give them out to the ongoing GWAS projects, we genotype the animals, and then we can select from those 500 animals that are predicted to have extreme phenotypes. And we can do that for any of the phenotypes that we've previously studied, but it's important to understand that we can't do that for a phenotype that nobody has previously studied. We have to have training data from some GWAS-like project to be able to do that. And so I have these slides here. It's not even in presentation mode. Terrible. Sorry. No, it's just very slow. Okay. Yeah, it's fine. Just a little bit of a lag. So I have here a partial list of some of the traits that we're good at predicting. And locomotor body weight are ones that we've gotten from lots of different labs. So we have really huge sizes. Everybody should be out of date now. And then we have a lot of other traits, which are mostly behavioral traits, study, where we have thousands or hundreds of animals. And I'm listing the sample size there that's available as training data, and then also the heritability of the trait. So the SNP heritability of the trait, which remember is different from the kind of heritability number that you might see if you did a panel of inbred strains, which is another way of deriving the heritability. But the SNP heritability is important. Higher heritability is going to mean better prediction. And it's a little bit out of order, but here is the figure that kind of shows that. So on the left, we're looking at 4 different traits. The y-axis is how well we can predict them. The x-axis is the sample size available for training. So we have big samples, so that meant we could downsample it to see how well our prediction would work if we pretended we had less data. And then the different colors are 4 different heritabilities that they're shown there. So kind of 0.13 up to 0.47. So 0.13 is pretty low to us, and 0.47 is as high as it ever gets for us. And what you see is that more data is better for prediction, and there's no evidence really plateauing there. It would be better if we had even more data. And higher heritability is better, of course, because how can you predict if it's not heritable? But even at around 500 is the number I have thrown around, our prediction is starting to get pretty good. Oh, he's kind of cutting out. Sorry, guys. Okay, let me stand over here. Where does the microphone? Is the microphone on this computer? Yeah, I'm sorry, guys. I forgot about that. Okay, that's much better. All right, thank you, Evan. Okay. So we can predict single traits. That's sort of what we originally imagined doing. But we can do some other things too that I didn't mention yesterday. One of the obvious ones is we could predict many different traits. And so we have a group right now that's requesting animals predicted for 2 different traits. And we're sort of going to give them animals that are high for both trait 1 and 2, or low for both trait 1 and 2. And you can imagine we could do high for something and low for something else, or et cetera. So you can do multiple different traits. We can predict on something as simple as a single SNP. So we have one collaborator that had a GWAS finding, and they're interested. They think it's a metabolic enzyme that's actually helping to metabolize cocaine more or less quickly. And so what they wanted to do is get some animals that were selected just for that SNP that would be the high metabolizing or the low metabolizing variants, they're going to give them cocaine and see if they actually metabolize it faster or slower. So basically testing that hypothesis. So that's an example of where predicting on a single SNP could be useful. One thing that we could do, and actually when you were talking about models for rats, we can predict for thousands of genes where there are pretty strong cis-EQTLs, we can predict expression of a gene. And somebody that wanted something kind of like a knockdown or an overexpressor, we could give you animals that are predicted to be homozygous for the high or the low expressing EQTL variant. So if I'm just by memory, there's about 1,000 genes where there's about 30% heritability to the gene expression, and I think more like 4,000 where there's 10% or more heritability to the gene expression trait. So another thing we could do is we could provide people with models where one gene would be differentially expressed, but no other genes should be systematically differentially expressed. And that's like a cheap way to get something that ordinarily would be expensive to make. Nobody's actually used that yet, but I think it's sort of a cool application of this. And that might be predicted on as little as one SNP, but sometimes there's a few SNPs that are good for predicting a gene's expression because maybe there's more than one cis-EQTL. And we're actually working with somebody to do trans CQTLs also. So maybe in the future, we'd be able to try to predict trans effects, which different people have different religions and belief in trans versus cis-EQTLs differently. But I don't talk about religion. And we can also do coding SNPs. So there are a number of genes that have coding SNPs, including I think a few hundred that are predicted to be like loss of function level coding SNPs. So if your gene happens to be one of those genes, and again, we could display that on RGD if you wanted to, then we should be able to give you animals that would be like homozygous loss of function or homozygous wild type. And if you happen to be lucky and that's one of your genes, that could be a cool application. Again, we haven't done that. And the other thing that I've thought about, but that we haven't done, is it might be interesting to have animals that had predicted expression profiles over many, many genes. So for instance, Greg, whose talk I didn't fully understand, but Greg was coming up with a matrix of the trans signal or the non-cis signal for lots and lots of genes. And so if you thought you knew what the gene expression profile looked like, say for case and control, and you might have borrowed that from a human study where you thought you had a differential gene expression profile across many genes, we could produce animals that were as close to that profile as possible just by taking into account lots of different cis-EQTLs for lots of different genes. And maybe there's a future in that, in coming up with animals that have sort of disease-like or non-disease-like gene expression profiles across lots of genes. And that's something that I think is a cool possible application for this we haven't done yet. So just to recap, Radica, this funny thing where we are producing a lot of animals anyway. It's not that hard, well, it's not that easy, but it's not that hard for us to genotype those animals when they're very young, so that by the time they're 50 days old, which is sort of young adult, about the time you would ship them out, we will have genotypes and we can then predict things about those animals and send people animals with predicted traits. And one of the real powers of that is, especially for, say, some of the exposure-based traits that people study, people that we work with study, like cocaine self-administration. If you get animals that have already taken a lot of cocaine and already didn't take very much cocaine, they might be different because of the predisposing thing that caused them to take cocaine or not cocaine, or they might be different because they've now taken a lot of cocaine or not very much cocaine. And those two things are perfectly confounded in that sort of a situation. So what Radica can offer there is we can give you animals that are genetically likely to take a lot of cocaine or not take cocaine, but none of them have ever seen cocaine yet. So now you can really look at what is the phenotype, and it might be a gene expression phenotype, it might be a circuit phenotype, an electrophysiological phenotype, whatever. What is the phenotype of an animal that's at genetic risk for some behavior like that, even though they've never engaged in the behavior? And again, it's sort of hard to think how else you would do that other than say selected lines. Somebody's out there. Okay, so that's what Radica is. We're doing it tomorrow. I'll try to bring stickers. I forgot, but we have some like Radica stickers and things that people think are fun. And so I'll try to bring some of those tomorrow morning for you. And we're really interested to get feedback about this. We would love users, people that want to work with us on this. And oh, and the other thing I was supposed to show you, sorry, but one other thing I was supposed to show you was, there's a web page about this. If you Google Radica, I found that this comes up right away. I actually noticed that ChatGPT has read our stuff about Radica, and if you ask it about Radica, it perfectly describes what Radica is, which was gratifying. But so here's the page that has more information about it. It's got a bigger table of things that we've... What's wrong? Oh, it's not sharing? Oh, geez. Okay, sorry, guys. I gotta stop share and reshare. Yeah, that's all right. I just wasn't thinking. Yeah, it's just that I thought I could change windows and have it do that. Didn't work that way. Okay. Here is the Radica page. Again, if you Google Radica, I think this will be the first result, or you could remember that URL. And here we have a request form where you can request Radica animals. It's just like a little Google form. And here's a longer list of traits that we think we're good at predicting, not exhaustive, but a longer list that we think is maybe more attuned to what people are looking for. And there's some more information here. We have a bioarchive that we really need to deal with and get published, but we have a bioarchive that's linked to here also that describes sort of how we do this. So that's what Radica is. We would love feedback about this. Is this useful? Do you have ideas about what you can do with this? Do you want some of these animals, etc.? Okay, that's it. Thank you.
One question, and we're actually gonna take this question and then we're gonna push through because we're on break and we have virtual people are online.
Oh,
For the
no.
next talk, so I'd like to just push through those and we'll break after the virtual talks, if that's okay.
Yeah,
Real question.
yes.
So you distributed graph.
Yeah.
When you take the data back, like you collect the data.
We don't need to. I mean, I assume that people are doing this because they want to publish a paper, and we'll certainly work with them to do that. But you know, we're giving them the animals, and they wouldn't need to necessarily include us in that. So it could work either way. But you know, the nice thing about it is it's basically a two-group comparison, right? You've got high and low, and you can do a t-test to see if they were good. That makes it really accessible, I think, to people more geneticists. Okay. Yeah.